Welcome back, budding biologists, to your video notes. Today we are going to end our unit on viruses by discussing the topic of viral replication. We will explore the two key processes, the lytic cycle and the lysogenic cycle, and essentially we're going to learn how viruses hijack host cells in order to reproduce and the differences between our two cycles. All right, let's make it make sense. All right, by the end of this video, you should be able to compare and contrast the lytic and lysogenic cycles and determine whether a virus is lytic or lysogenic based on a given scenario and any symptoms. We have two types of viral replication. That's going to be the lytic or lysogenic cycle. Lytic replication results in viral particles being replicated and then rapidly destroying their host cells. It's a short, quick cycle. Lysogenic replication results in the viral DNA being integrated and becoming part of the host cell's DNA. It does eventually lead to cell death, but it's a little bit longer. Right, let's talk about our fundamental differences between these two cycles. All right, let's focus on exactly how viral replication happens, starting with the lytic cycle. All right, to illustrate our cycles, we do have bacteriophage as our example over here. So here's our little phage guy. And it is important to remember bacteriophages do specifically infect bacteria. We're just using it as an example, but keep that in mind, please. All right, talking about the stages of the lytic cycle. There are 5 key stages that describe how the virus infects the host cell. Number 1 is attachment. The bacteriophage is going to first recognize and attach to the host cell. So over here, he's attached to the host cell. Think of this as the virus finding the lock that fits its key. Remember, it follows that lock and key model we discussed earlier in our last video. Once attached, the virus injects its genetic material, which is typically DNA but can be RNA, into the host cell. This is similar to a needle injecting its contents into a target. We're just injecting the material. The actual bacteriophage is not going to go into the cell. We're just injecting the genetic material inside of the cell. Number 3 is replication of viral parts. Once inside the host cell, the viral DNA takes control of the cell's machinery. It hijacks the cell's ribosomes, enzymes, other components to replicate its DNA and produce virus-specific proteins. Number 4 is assembly. The virus is going to use the host cell's machinery to make new viruses using the replicated DNA and synthesized proteins. And number 5 is release of new viruses. The host cell eventually becomes overwhelmed and bursts open. That's what that word lyse means. It's going to burst open due to the sheer number of virus particles that are produced. This event releases multiple viruses, which then go on to infect other cells, perpetuating this cycle. It's going to start all over again with a new cell from those new viruses. One crucial aspect to note about the lytic cycle is that symptoms of infection occur very rapidly. This is quick, quick, quick. This is because the host cells are destroyed and a significant number of new viruses are released in a very short period of time. Examples of lytic viruses are the common cold and flu. When these viruses infect a host, they take a much more aggressive approach to replication. The quick replication process leads to those hallmark symptoms of the common cold and flu: coughing, sneezing, congestion, fever. Typically, symptoms occur within days of infection, and once infected, the host is unable to treat the virus. You can try to prevent it if a vaccine is available, but once infected, all the individual can really do is manage the symptoms. Typically, this includes getting plenty of rest, drinking lots of fluids, and allowing the body to recover. Eventually, the immune system will kick in and take care of that pesky virus. Here's another diagram of the same process. Please jot this down in your notes. All right, moving on to the lysogenic cycle. We once again have our bacteriophage over here as our example. In the lysogenic cycle, there are 3 key stages that you need to know. So number 1 is injection. We're starting here this time. Similar to the lytic cycle, the virus first attaches to the host cell and injects its DNA. This initial stage is identical in both cycles. Number 2 is integration. This stage is where the lysogenic cycle differs significantly from the lytic cycle. Put a star next to this, please. Instead of immediately taking over the host cell's machinery, the viral DNA integrates into the host cell's DNA. It becomes part of the host genetic material, forming what's known as a prophage. Number 3, latency. During latency, the prophage remains dormant within the host's DNA. The integrated viral DNA doesn't actively produce new virus particles, and no symptoms of infection are observed during this period. Instead, the viral DNA becomes a permanent part of the host cell's DNA. It becomes a sequence of genetic material that is replicated along with the host's DNA when the cell undergoes cell division or replication. During this time, the infected organism likely will not have any symptoms and may not even know they're infected. This virus can lay dormant for extended periods. It may be days, weeks, months, or even years before symptoms occur. If external factors such as heat, exhaustion, exposure to chemicals or radiation, etc., stresses the host cell, the virus can be triggered and exits its dormant phase. If this happens, the virus will remove itself from the host cell's DNA and initiate the lytic cycle to go through replication, assembly, and release, which causes the host cell to be lysed and several new viruses to be released. Keep in mind that the lysogenic cycle's unique feature is its dormant phase, where the virus remains integrated within the host DNA, as opposed to the rapid onset of symptoms in lytic viruses. A prime example of this is HIV as well as herpes. I'm going to use herpes simplex virus 1 or HSV1 as an example. Remember from our last video, it's estimated that 67% of the world population is infected with HSV1. Upon infection, HSV1's genetic material becomes integrated into the host's DNA. It's like the virus blends into the host's genetic code, becoming part of it. And HSV1, also known as oral herpes, can remain dormant within the host's DNA for extended periods, sometimes years. During this dormant phase, our latent phase, the infected individual typically experiences no symptoms. It's as if the virus is hibernating within the host's cells. Remember, though, HSV1 has the potential to reactivate if it's triggered. This is when symptoms become evident and the individual will develop cold sores or fever blisters, typically around their mouth. An individual with HSV1 has a lifelong infection. There is not any treatment, so the infected individual's options are to take antiviral meds to reduce the severity of the outbreak, manage symptoms with creams or over-the-counter medications, avoid close contact with others during an outbreak to prevent spreading the virus. But the HSV1 virus will forever exist within their cells, and they'll likely cycle between periods of dormancy, where they don't have any symptoms, and then on occasion have an outbreak of cold sores or fever blisters. Here's another image. Please jot it in your notes. All right, let's practice identifying whether an infection is lytic or lysogenic. Deep in the Mariana Trench, marine biologist Dr. Alexander Corbett and their team are exploring the world of extremophilic archaea. They collect samples from the ocean floor, hoping to understand how certain organisms adapt to extreme conditions. However, a mysterious viral presence disrupts their research. The samples reveal a swift decline in archaea populations and a growing number of viral particles. Dr. Corbett and the team race against time to salvage their research amid this sudden ecosystem imbalance. Based on this scenario, what type of viral replication cycle does this virus seem to undergo? Be able to explain your reasoning. Pause the video right here and take a minute to think about it, please. All right, based on the scenario, it appears that the virus in this situation is undergoing a lytic viral replication cycle. Congrats, pat yourself on the back if you got that right. The text evidence is number one, firstly, that the scenario mentions there is a swift decline in archaea populations as a result of the viral presence. So, this decline suggests that the virus is actively infecting and destroying the archaea cells. Your second point of evidence could be that the scenario notes there is a growing number of viral particles. That indicates that the virus is actively replicating within the archaea cells and producing new viral particles, which is characteristic of the lytic cycle. And then lastly, you could have said that the research team is racing against time to salvage their research due to the sudden ecosystem imbalance. That supports the idea of the lytic cycle. Remember, in the lytic cycle, viral replication is rapid and it leads to the destruction of those host cells. So if you see anything about rapid onset of symptoms or the word sudden in any context, that's a clue that you're likely looking at the lytic cycle. All right, guys, I hope this helped. I am going to wrap it up here. Please jot down any questions you have over our unit and schedule time at tutoring if you're struggling. Otherwise, I'll see you guys in class.